Hey everyone, welcome back. So this is our tissues lecture from module 3. At the end of this lecture, you'll learn about the 4 primary tissue groups and all the subgroups that belong to each of the primary tissue groups, and then which type of organs are formed by these tissues. Okay, so we're going to be taking a look at the epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Now, these are the 4 primary tissue groups, and each one of these primary tissue groups are then going to have subgroups that belong to them. We'll also learn about coverings and membranes, so serous membrane, parietal membranes. We'll learn a little bit about how tissue repair happens and then go through the developmental stages for tissues. Okay, let's get started. So what are tissues, right? Tissues are groups of cells that are similar in structure, not all of them, most of them, but they're similar in structure and they perform a common or related function, right? So that they have a similar goal, and so they work together, okay? There are 4 main tissue groups, also called primary tissue groups, and the 4 primary tissue groups are going to be epithelial, connective, muscular, and nervous, okay? So these are primary tissue groups, and we're going to take a look at each of the primary tissue groups and look at the different characteristics that make it that tissue group, and then all the subgroups of tissues that are categorized within that primary group. So we start off by looking at epithelial tissue. So epithelial tissues are sheets of cells. These are cells that are connected together tightly by adhesion proteins. Remember, we learned about that in the last class. So they form these sheets of cells that, what do they do? What does a sheet do? It covers, right? So it covers the body surface. It'll cover or even line the body cavities, okay? There are 2 types of epithelial tissue. One is the tissue that are sheets of cells, and those are going to function as covering and lining. And there's a modified epithelial tissue that are glandular. So they form glands, and so those cells are going to produce and secrete chemicals. Okay, and we'll see what those are coming up. Okay, so as you can see, this is just a transverse section through a body, and all this, I just put this here so you could see all the different, all these little white lining that you see, all of that is epithelial tissue covering the outside of organs and also cavities. So what are the characteristics of epithelial tissue? One, as we've already said, the epithelial tissue cells are tightly packed. They're tightly packed next to each other. There's very little acellular space between the epithelial tissues. And this is specifically for the sheet-like function of epithelial. Remember, there are 2 types of epithelial tissue. One, which are sheet-like cells, and their function is to cover and protect. And the other type of epithelial tissue, a modified epithelial tissue that are glandular, right? So they look different, they function differently, okay? So when we talk about composed of tightly packed cells, we're talking about the first type. We're talking about the sheets of cells of epithelial tissue. Adjacent epithelial cells are bound together by specialized proteins called desmosomes and tight junctions. Again, this is not the case for glandular epithelial. Epithelial cells exhibit a polarity, right? So don't think about polarity as we've been talking about up until now as in having a charge, but polarity in this sense means that they're different ends, okay? So there is an apical end, which is free, and then a basal end that's attached. So what does that mean? When we're talking about epithelial cells, for the most part, they're going to be coverings, right? So if you're covering the inside, right? So let me see if I can. Right? So if you're covering a inside of a cavity, right? If this is the covering, I should have made a different color. But anyway, this is a covering, and this is a cavity or the lumen, right? Lumen means opening or cavity. Cavity means space, right? Like inside your intestine, the tube, there's going to be space inside the tube, right? So the cells are going to be like this. I'm not going to draw all around because that will take forever. Okay, so there's this end on the cell that faces the open lumen space, right? And then there's the other end, right? The other end that's not, that's attached to the organ, right? So it's attached to the organ. So that's what we're talking about, that there is a polarity. Okay, there's a polarity where there's a end that is free and not attached, and that is called the apical surface. And then the other that's attached, and that is the basal surface. Okay, so this is the apical, right? Because it's free, it's not attached to anything. And then there's the basal that's attached. Okay. The epithelial cells are innervated, which means there are neurons that are present there, but it is avascular, which means blood vessels don't intertwine in between, capillaries don't intertwine in between the epithelial cells. The blood vessels are in the material that the cells are attached to, right? The material that the basal surface is attached to. And we'll see what that material is coming up. And epithelial cells have a high regenerative capacity, meaning they go through mitosis often. It makes sense, right? Because if they're lining the inside of cavities, what happens in cavities, things are passing through. So there's a lot of abrasion, right, of movement. And so these cells have to be highly regenerative, otherwise it wouldn't, serve its purpose to protect, right? If they die off. The sixth characteristic, again, as we said earlier, the epithelial cells have a polarity, right? They have a, oops, trying to change the color. Okay. Okay, so this would be the lumen. Okay, right. So this is the apical surface, the free surface, and then this is the attached surface, just like we said. And that's the basal surface. So what is this basal surface attached to? It's attached to material that is acellular, meaning it's not made of cells. This material that the basal surface is attached to is called the basement membrane. Basement membrane. Okay, so the basement membrane is acellular material. It's just fibers created by connective tissue, and underneath that then would be connective tissue. Underneath the connective tissue would most likely be a muscle, either smooth or skeletal, depending, of course, and where we're talking about. Okay, so epithelial cells lie on that, or the basal end is attached to the basement membrane. The basement membrane is composed of acellular material. Okay, so it's just fibers. The basement membrane you can think about it as that sticky layer that the epithelial cells attach to. Okay. Again, the surface that attaches to the basement membrane is called the basal surface. And the other end of the cell that is free facing the lumen is called the apical surface. Okay, so here are some pictures and diagrams to help, right? So these are our epithelial cells. Notice how they're tightly held together via these tight junctions. And this is the apical surface. Here's the lumen, which is the opening in whatever cavity that this is lining. And this is the basement membrane, right here. And the basal surface would be on this end here. And so actually this arrow should be, well, it's fine. So the pink line there would be the basement membrane, and this end of the cells would be the basal surface. And this is just underlying connective tissue that they're showing. So let's look at now the functions of epithelial cells or tissue. So one, when we're talking about the sheet-like function, remember there are two functions, right? One, the sheet-like function, and the other is glandular. When we're talking about the sheet-like function, or the sheet-like epithelial cells, or tissue rather, their function is protection, right? Because it forms this barrier from the outer world to the inner cavity spaces. Glandular epithelial main functions are for secretion, right? So these glands are going to secrete chemicals, and those chemicals can be part of the exocrine system or part of the endocrine system. Okay, so they secrete chemicals like hormones or enzymes to break down food particles within the digestive system, of course, right? They also have a role in absorption. So epithelial cells can also absorb materials. So where would you need absorption from? Digestive system, right? So lining the stomach, lining the small intestine, large intestine, are epithelial cells, because they're going to absorb those food monomers that we just talked about in our metabolism lecture, right? Are food monomers that are released, and they're going to absorb them and pass them on onto blood. Okay, also have a role in excretion. And so where would these be? Clearly in our excretory tracts. So urinary tracts, so also in the kidney, also in the tube that that urine passes through. And so that's the urethra. Okay. So let's take a look at now how epithelial cells are classified. Okay. So they're classified based on, one, their shape, and two, they're based on how many layers of the cells there are. Okay, so they're different shapes, and then but there are only two ways they can be layered, either single layer or multiple layer. Okay, so if it's a single layer, they're called simple, and if they are multiple layers, they're called stratified. Now, pseudo-stratified, so pseudo means fake. So pseudo-stratified means that it looks like it's multiple layers, but it's not. Okay, so you may say, well, why even call it that? Why not just call it simple? Because it looks different. The pseudostratified cells look different from the cells that are single layered. Okay, so let's take a look at what these different shapes are. Or, well, let's first look at the layering, right? So simple means there's only one layer of cells. Stratified is when there are multiple layers. And here's our pseudostratified. So notice how the pseudostratified, there's only one layer. Now, the simple columnar and the pseudostratified columnar that you see here, they're both column shaped, but look at the difference. All the nuclei of the simple columnar kind of line up, right? But look at the nuclei of the pseudostratified. They're all at different heights. Okay, so they look different. Okay, so that's why they're called pseudostratified. Okay, so now we take a look at the shape. So epithelial cells can be flat and amorphous, meaning not really having a set shape, and those are called squamous. Okay, then you have cells that are cube shaped. Easily enough, they're called cuboidal. And then there's cells that are shaped like columns, and easily enough, those are called columnar. Okay, so you can have simple squamous or stratified squamous, simple cuboidal, stratified cuboidal, simple columnar. You can have stratified columnar, but not as common. And then, of course, our pseudostratified. Okay, this is a great slide, or a great table to study off of, because it gives you a good snapshot of everything for each of these different types of tissues, epithelial tissues. Now we'll take a look at each one of those cells. So let's take a look at simple squamous epithelium. Okay, so simple squamous epithelium is going to tell you that simple is going to tell you it's one layer. Squamous is going to tell you that it's flat amorphous. And then, of course, all of the other characteristics of squamous epithelium. So the main function, I'll start off with functions, because that's going to make sense more of where it is then. Okay, so because they're just very thin layer, simple, right? So it's one layer, and they're flattened cells, so they're not very protective, but they can help facilitate filtration, diffusion, osmosis. So they're a good in between layer. They're thin layers, and so they help. Oh, there's something in my hand. It helps keep things from drying out, right? So they're not really meant to be protective, but it does have a covering, provide covering, and it provides some kind of a thin barrier, but their main functions are going to allow for filtration, diffusion, and osmosis. So where would you find them? You'd find them wherever you need these kind of functions. So lining the alveoli in the lungs, right? lining blood vessels, lining the internal chambers of the heart, right? So these are going to be places where you would find simple squamous, simple cuboidal. So I just wanted to point something out. So here is a actual histology slide on this side, and on this side you're going to see the diagram rendition. So this is what these are going to look like. Nice and flat. The nuclei are not really localized in a specific space from cell to cell. And the nuclei are, wherever it is. They're, as you can see, they're tightly packed, and so this is, and they're pretty thin. Cuboidal cells, so here is cuboidal cells, and they're so beautiful. They're cube-like shape. They have a large kind of centrally placed nucleus. And so you can see how this is a cube-like cell, right? So here's the diagram for it. And so you can see very distinctly they're cuboidal cells. There's only one row of them, right? And so these are simple cuboidal. So what is a function of simple cuboidal? So simple cuboidal is going to be secretion and absorption. And really, for the most part, their main function is for secretion. You'll find cuboidal cells are what are the cells that make up the different glandular epithelial cells. They do have a function in absorption, but really their major function is for secretion. So where would you find cuboidal cells? Wherever you need a secretive function, right? So wherever you need glands. So kidney tubules, ducts of glands, covering surfaces of ovaries, really wherever you need secretion of endocrine or exocrine function. Now we look at columnar. So if you take a look, let me see if I can go closer, right? Get some annotation out, right? So here is a nucleus, right? And if you follow the membrane, you could kind of see, right, here's another one, right? So you can see they're column shaped. And at the very top here, what you're seeing are microvilli. And we'll see more about microvilli when we learn about the digestive system, right? So these are simple columnar, and this is a lumen. And so obviously this is the free end. This is the basal end here. This is the basement membrane that you're seeing here. Okay, this is the nucleus. Okay, and these are column shaped. Now, if you notice in between these columnar cells, and these like ghost-like cells, and those are called goblet cells. Okay, goblet cells. So goblet cells are modified epithelial cells that produce and secrete mucus. Okay, so that's what. Okay, so columnar cells, so simple columnar cells, and you can see them in the diagram. They're column-like shape. Notice all the nuclei are kind of lined up, right? They're all lined up towards the basement membrane. So the main functions of columnar cells are, secretion and absorption, but really their main function, unlike the cuboidal cell, is absorption, right? I mean, they're shaped to be more absorptive, right? There's more space in there for them to absorb materials than cuboidal cells. So the main function for our columnar cells is absorption and also protection, right? Because they're so tall, if you will, or long, that it does have a protective function. So where would you find columnar cells? Well, wherever you need absorptive functions. So GI tract, ducts of glands, and places like that. So those are our columnar epithelial. And columnar epithelial are going to have interdispersed in between them these modified epithelial cells called goblet cells, and goblet cells produce and secrete mucus. So now we take a look at pseudostratified, right? So going closer. So notice how the nuclei are all at different heights. So really you would think, oh, this is one layer and here's the other layer down here. But these are just single cells. So if we highlight, I show you. So here is, and this is the cilia that are sticking out. And these are ciliated pseudostratified epithelial cells. Let's see if we can find another one. Okay, so these are simple pseudostratified columnar epithelial cells. Okay. Okay, and then of course you have the diagram on this side. Okay, so these are pseudostratified and these are ciliated. So ciliated cilia are these hair-like structures that stick out from the surface of cells. And what do they do when they move around, right? When they move around, they move the fluid and move the components that are around it in different directions. Okay, so the function for the pseudostratified are protection, secretion, and movement of mucus. Okay, and movement of components. So, and then another example would be sex cells. So, where would you find ciliated pseudostratified columnar cells? Well, wherever you need to have to keep things moving, right? So, in the fallopian tubes, right? The ovarian tubules to move the egg along. You would find it in the vas deferens, right? From the testicles to move sperm along, right? You would find them in our trachea to move, particles of dust and things out of the trachea and into the esophagus, right? So where would you find it? Lining the respiratory tract, part of the reproductive tracts, okay? And so these are the pseudostratified ciliated columnar epithelial cells. So now we take a look at stratified epithelium. So we're starting off with stratified squamous epithelium. So stratified squamous is that squamous cells that have multiple layers. So where would you need to have multiple layers of cells? Clearly in areas that you have a lot of movement, right? A lot of movement, so there's a lot of abrasion, so that you need to protect the underlying tissue. So you can have this thick layer of stratified cells that can get sloughed off, but it's not going to hurt the underlying tissue. So the function for stratified squamous is protection from abrasion and desiccation, right? So we'll talk about keratinized when we learn about the integument in the next lecture. But keratin is a protein that's fibrous in nature, or it's fibrous, and then when they are next to each other, they kind of just stick together and form this net. Like if you can think about a bunch of hay strands, they stick together, and when that happens, when keratin is in cells, it actually kills the cells, but it also forms this kind of waterproof barrier, and we'll learn more about that when we talk about the integument system. And so where would you find it? Wherever you have lots of movement, so lining the esophagus, mouth, vagina, anus, and also on the epidermis. Okay. And also places like the tongue, right? So this is actually the tongue. And so these are stratified, you can see it, right? Stratified squamous epithelial cells. How do you know that they're squamous? Well, because they're flattened cells. They don't look column shaped and they're not cuboidal in shape. So they are stratified squamous epithelium. And this is an example on the tongue. And then you have transitional epithelium. So transitional epithelium is called transitional because the cells change shape from the basement membrane to the top layer. So number one, transitional epithelial are stratified cells. They're stratified, and the shape of the cells changes as you get to the top of the towards the lumen, right? So the transitional epithelium resembles both stratified squamous and stratified cuboidal. Okay, the basal cells are cuboidal or columnar. So the basal cells here tend to be stratified or columnar in shape, and they become more squamous-like as you get towards the lumen. They kind of flatten out as you get towards the lumen. So what that allows is stretching of that tissue, right? It allows stretching or distension of that tissue. So where would you need this kind of tissue that allows for distension? Well, wherever you're going to have organs that are used for storage, right? If you're going to use something for storage, you need to be able to, for it to expand. So part of the ureters, which are these tubules that come out of the kidney that carry the urine towards the bladder, right? And then, of course, the bladder itself, right? And then the tube that carries the urine out, the urethra. Okay, so wherever you need this kind of, these are just examples of where it's not the only place, right? So in your textbook, we'll show you different places that you would find transitional epithelium. And so this is a great pictograph, or micrograph rather. Okay, so notice here they're more column shaped, right, towards the basal end, even cuboidal shaped here. And then as you get towards the top, they kind of flatten out and become more squamous like. So now let's take a look at glandular epithelium cells. So glandular epithelial cells are different from the sheet-like epithelium cells in that they are not tightly packed, right? They're kind of like little islands of their own, little glands of their own. And so they consist of groups of epithelial cells that have been modified to synthesize and secrete specific chemicals. Glands can either be exocrine glands, so exo means out, right? So exocrine glands secrete their chemicals onto an organ or in an organ through a passageway. So it's directly deposited onto or into an organ. Okay, so excretes, so this not excrete, secretes. Exocrine glands secrete to the external environment via ducts. And external environment doesn't always have to be out here, right? External environment meaning into the outside of the gland, right? So into or onto another organ, okay? The other type of glandular epithelium are endocrine glands. So endo means inside, right? So endocrine glands secrete their chemicals that get picked up by blood, and those chemicals travel through the circulatory system. And these are ductless glands, okay? A duct is a passageway. So the endocrine glands are not attached to a duct because they just secrete their chemicals into the interstitial fluid, and from there it's picked up from blood. Okay, and then you have mixed glands, and these are glands that have both an exocrine and an endocrine. Not that the same gland does both, it's just that this organ will have both exocrine and endocrine glands. And a great example is a pancreas. So the pancreas has exocrine, the pancreas has exocrine glands that produce digestive enzymes that flow through the pancreatic duct and enter, then released onto the small intestine, specifically the duodenum, which is the first part of the small intestine. And then pancreas also has endocrine glands that produce and release two hormones, namely insulin and glucagon. Okay, so that's a mixed gland. So we will learn about exocrine glands in this lecture. We have a whole lecture for endocrine, and that's further down the line. So we're just going to focus on exocrine glands right now and exocrine secretion. So exocrine glands have ducts, and they secrete their product onto the surface or into body cavities. So what that means is, or what that looks like is this, right? So here's the gland, and it's going to produce its products. Those products are passed through a passageway called a duct, and the duct is then going to open up into wherever those secretions have to go. Okay, so this would be the factory, if you will, and this is then, the passageway from the factory to bring the goods to where it needs to go. Okay, so this is called the secretory unit, because that's where it's going to, all the secretion and production occurs. This is the duct. Okay, and then of course, this is just the organ or the cavity in which the material is going to be secreted into or onto. So these exocrine glands can be either unicellular, so there's just one cell that does everything, or multicellular, where you have, more than one employee. Okay, so there are three types of exocrine secretions. merocrine, apocrine, and holocrine. Okay, so let's take a look at the unicellular, multicellular, and then we'll learn what merocrine, apocrine, and holocrine is. Okay, we'll talk about the different structures and shapes of the exocrine glands, whether they're unicellular or and multicellular. First, we'll talk about this. I switched it around. I should switch it around from here. So I'm talking about these glands, and these glands that, and then they release their products into ducts, right? But the way that secretion is released is different. There are 3 different ways that the exocrine secretory cells are going to secrete their chemicals. Okay, so here's the basement membrane and the duct, and here's an epithelial exocrine cell, and here's its nucleus, yeah. And then in it, it's going to have the granules or the vesicles, right, that have the secretory chemicals, or the chemicals. The secretory cell, because it secretes these chemicals. Now, how are you going to get this green vesicle to release its chemicals into this open space here, right? This is the lumen. Okay. So you can say, well, maybe there's openings, like our channels that we talked about, and there are openings and the vesicle can squeeze through the opening. Or maybe there's a door that opens up on the vesicle and the chemicals just kind of diffuse out, right? Because there's more of that in here than outside. Maybe the cell has a door and the whole vesicle comes out, right? So all of those are valid ways of thinking about it. So merocrine, also called eccrine secretion, is one way, is one mechanism of secretion. So when I ask you, provide the mechanism of excretion for such and such gland, what I'm asking you is how does that secretion occur? Okay, so mechanism of secretion is going to either be merocrine, apocrine, or holocrine. What that means is, how is this chemical released? So there's a big difference between how something is released and why something is released. So when I ask you the mechanism of action for the release of a certain chemical of a certain gland, I'm not asking you to tell me why that's being released. Why would be what's the stimulus, what's the purpose? Mechanism of action is how it's being released. How is that happening? And the 3 different hows are going to be merocrine, apocrine, and holocrine. So different exocrine glands use a specific mechanism by way to release its granules, or the contents from its granules. Okay, so merocrine is one mechanism of action, or mechanism of excretion, mechanism of secretion for this gland. Okay, so the most type of exocrine secretion is via eccrine secretion. Okay, what happens is the secretory granules are going to travel to the plasma membrane, and because remember, these are vesicles, they're covered by they're covered by membrane, they're organelles, they're membrane bound, or eukaryotes. So but this membrane in green is the same membrane. It's phospholipids, like as the red plasma membrane. And so what's going to happen is it's just going to fuse. It's just going to fuse, and the chemicals are released. Okay, so the secretory granules, which were packaged in the Golgi, remember, go back. If you don't remember right away what Golgi is, this is a good time to stop and either write yourself a note, go look up Golgi, or go look up Golgi right now and then come back and continue. Okay, so these are packaged in Golgi bodies, and the vesicles migrate to the apical surface of the cells. The membranes of the secretory granules fuse with the apical membrane, just like how I drew, and the release results in the secretion of the components into the external environment. And this is just exocytosis. Okay, so if exocytosis is something that you're not familiar with, now is a good time to stop and go look up exocytosis. Okay, so one mechanism of secretion, how secretion occurs from an exocrine cell, is through eccrine or merocrine secretion. What is that? It is the fusing of the vesicle with the plasma membrane, opens up, and the contents then are released. So what is another way of secretion? The next is called apocrine. So remember, our cells, remember our epithelial cells have a free end that's called the apical surface, and then the basement surface, right? The basal surface. So this is basal surface. Okay. So if I say this is called apocrine, you know it's got to do with something with this free end that's facing the lumen. And so that's exactly what it is. So in the prior with eccrine, here was our vesicle, and the vesicle traveled to the surface, fused with it, and the contents were secreted. With apocrine, the vesicle travels to the plasma membrane, but instead of fusing, it kind of pinches off and is released. And then when it pinches off, it takes a little bit of the plasma membrane with it. It's just some detail, but anyway, right? It pinches off. Once it's out into the environment, the vesicle then just opens up because it can't maintain the membrane, it opens up, and then its contents are released. Okay, so the apocrine secretion is where apical portion of the cells, together with the secretory granule content, are budded off, releasing into the lumen. So that's just called budding. Okay, so if you don't know what the budding is or what budding is, stop now and go look it up so that you can understand this process better. Okay. Examples of apocrine secretion is found in sweat glands of armpits. We'll learn about where the different types of glands exist. Examples of eccrine glands would be sweat glands that are on our forehead. Okay. No, okay, it's okay. Then the last is holocrine. So far we've learned the vesicle travels to the surface, it fuses with it, and then the contents are left. Or we have the vesicle travels, comes off, and leaves. And now the only thing left is for the whole cell to leave. And that's what holocrine is, right? This is where the whole cell, along with all of its contents, is released. So here's our slant. Here's our cell. The whole cell gets released. Once it's released, it dies, right? It dies. Contents are open. All the vesicles open up, and the contents are released, right? Examples of holocrine secretion is on the sebaceous glands. Examples of where you would find holocrine secretion are in sebaceous glands. And we'll learn more about these glands when we learn about the integument in the next lecture. But it's important for you to understand mechanism of secretion now, so then when we're talking about these different glands with our integument system, you'll already know how this secretion occurs. Now we talk about, remember earlier we said that there are unicellular and multicellular exocrine glands. So let's talk about unicellular first. So unicellular, we've already seen a unicellular exocrine gland, and that is our goblet cell. Okay, so it's just one cell, and it produces its mucus, and it releases the mucus, right? So these are dispersed throughout the epithelial linings of the intestinal and respiratory tract, because that's where you need a lot of viscosity, because there's a lot of movement, right? So it produces a molecule called mucin, which is a glycoprotein. When it dissolves, it becomes water soluble when it's in water, and then when it does, it becomes viscous. like mucus. I mean, it's mucus, right? And so mucus is viscous, and so it is protective because it decreases friction. It's protective because it's thick, so it can trap pathogens and dust particles, right? And of course, it's lubricating and it prevents desiccation, which is drying out. Now we take a look at the multicellular. So multicellular obviously are going to be structurally more complex than unicellular. There are two parts to a multicellular exocrine gland. Okay, so one is the factory, right? It's the cells that produce and secrete their contents or their material, right? Those are called the secretory units, also called the acinus, right? And these are made up of cells that are modified to produce and secrete their specific chemical. And now you have to, ship the product from that factory to where it needs to go. And that pathway, right, is called the duct. Okay. So you have two parts to it. You have the factory, which is the acinus or the secretory unit, and then the route through which the products are sent through, and that's the duct. Okay. These exocrine glands are going to be further classified based on their structure and the secretory units. So you can either have, so these glands can be simple or compound, and then the shape of the secretory unit can either be tubular, so shaped like a tube, or spherical like or alveolar. So let's take a look. Okay, so in this, this is the, okay, so this is the secretory unit, and this is the duct. Okay, again, secretory units, and these are the ducts. Okay, so what's the difference between simple and compound? Simple means when you have one secretory unit that is connected to one major duct. Compound is when you have lots of secretory units that have their own duct that are then connected to a major duct. Okay, so in this case, there's just one pathway. And in this case, there are several units with their own little off ramps that are then going to then connect to one major duct. So that's simple, where it's just one secretory unit and one major duct. Compound, when there are several secretory units with their specific, their ducts that connect to one major duct. Now let's take a look at the shape. So the shape can be either tubular, as you see here, or roundish in shape. Oops, roundish in shape, like this. Okay, so tubular, or roundish in shape. So tubular, shaped like a tube, it's tubular. So this is simple tubular because it's one secretory unit to one major duct. This is compound. Well, actually, be careful. See how I just made that mistake? This is compound. So try not to think of compound as several secretory units and simple as one secretory unit. What the distinction between simple and compound is the duct. Are they all the secretory units sharing one duct, or do they have their own ducts that then merge into one duct? Okay, so these are several secretory units, but they all share one duct. Now, this are several secretory units, but they each one has its own duct, which then merges into one duct. Okay, same thing for alveolar. They're alveolar shaped, spherical shaped. Okay, so this is simple, and this is compound. So this is also simple, even though there are several secretory units, they all are sharing one duct. Okay, so simple duct, the duct does not branch. Compound, the duct branches. That's an easier way to think of it. Okay. I'm going to stop here. So this way, and then I'll have a separate connective tissue lecture recording, and then a separate muscle and probably nervous system recording. So I'll probably have 3 recordings for the tissue, so you don't have to sit through a whole hour of it. Okay, so I'm going to stop here, and the next one will be on connective tissue. Okay, that's it.